Some of this video will be review over material that you should have learned when you were in introductory biology, maybe even going back to high school biology. But I found that it's a good review, and also there are just some people that need the reminder or that haven't had it before. So we're going to talk a little bit about genetics, but primarily we're going to be looking at the function and characteristics of the DNA code. So first, Mendel did these very involved experiments where he was looking at the nature of how traits were passed down from one generation to the next, and he used pea plants. And so he chose characteristics that he knew he could easily measure in the next generation. So things like flower color, or like the shape of the peas in the pod, the height of the plants. And these are things that had very binary characteristics, so it was one thing or the other, and that he knew that he had true breeding populations for, so he could then breed them together and see what the offspring was like. And so through that and very, very careful records of the number of offspring that were produced by those, he was able to figure out how traits were passed down from one generation to the next, at least the kind of simple version. There are more complexities that have since been covered and discovered, but he's the one who found those initial characteristics of how those traits are passed down. Before Mendel, it was thought that traits were passed down through a blending, which makes a lot of sense if you don't look at all the details and if you don't do all these very specific experiments that Mendel did. So the idea of a blended inheritance is kind of like paint or like two dyes. You have a blue dye, a yellow dye, you mix them together, you end up with green. And so this made sense when you looked at offspring and thought, oh yeah, offspring look a little bit like mom, a little bit like dad. And so we have a new third entity that is made up of the components of both parents. Now, the problem with this, and this was one of the reasons why Darwin had so many criticisms, and he himself criticized his work about the amount of diversity that we see in populations. It was thought that if you have a blended pattern of inheritance, that eventually you would lose all the diversity. And think of it this way. Let's say you have 10 distinct, unique colors of paint, and you mix them together to create generation 2 of paint. And then you mix those together, just 2 at a time, but you mix those together and make generation 3 and so on. Eventually, all of your paint is going to be very, very similar, kind of a brownish gray color, and we would lose all the independent, distinct colors that we started with. Okay. However, Mendel realized that rather than being a blended model, that a particulate model was much better. So in this case, they've shown these little filters to be similar to their original example, but I like the idea of marbles better, right? So if you have a red marble and a white marble, you put them together, you have characteristics and qualities of both, but yet each has remained distinct and separate. Okay, so of course today we know that those independent particulates, right, we call them alleles. And this is important. We'll look at the vocabulary again at the beginning, but I think I want to mention this now. Oftentimes you'll see the word gene and allele used interchangeably. However, there is a big difference. So unless there is some major mutation, all humans have the same genes. But you'll see people say all the time that individual A is different from individual B because they have different genes. That's not the correct usage. What they really mean is alleles. So everyone inherits the same genes. However, we get different versions of those genes from our parents. So for instance, everyone has the gene that determines eye color. If you get a brown allele from your mom and a blue allele from your dad for eye color, then those two alleles will, depending on dominance, recessive, and a number of other factors that we'll talk about a little bit in this class, but that's mainly a topic for the genetics course. But if you have the two of those will together determine what your phenotype is going to be for eye color. So just be aware of that. When I say allele, I mean a version of a gene. And the genes themselves, everybody has those exact same genes. Okay, a gene is simply a piece of DNA that can be transcribed and usually translated and results in some impact on the organism, some phenotypic impact from that section of DNA. Okay, now we're going to review again. Hopefully most of this is review for you. Meiosis is the form of sexual reproduction that divides the genetic material in half. So meiosis is what's gone through before we make eggs and sperm or any other reproductive cell. Gametes, of course, are the result of meiosis. So eggs and sperm in mammals, there are other terms for, or eggs and sperm in animals, but there are other terms in different organisms. A locus is one you may not be familiar with. The name literally means location or address. So location is the physical place in a genome on your genes. You look at your DNA where we would find a specific feature. It could be a gene, it could be a microRNA or any other feature that we're looking at. So that's a locus. Law of segregation is, of course, what Mendel was talking about. It simply means that when you have two different alleles, that as meiosis occurs, those individual elements, those alleles, are randomly broken up into the gametes. And so this is also called independent assortment or random assortment. So you'll see all of those. Law of segregation is kind of the original term. Much more frequently, people say independent assortment or random assortment today. So just be aware that all those are interchangeable. And then, of course, we have this idea of dominance versus recessive. And Mendel was working with traits that were either dominant or recessive. And this simply means that if you have two different alleles, one of them will determine your phenotype, and the other one will be just kind of a placeholder. It's still there. It can still get passed down to the next generation, but it won't have an impact on your phenotype. Human blood type is a really good example of this. The O allele is recessive to both the A and the B allele. So if you are O blood type, I know that you have two O alleles, because that's the only way that you can show that phenotype, the O blood type. If you have an A blood type, for that matter a B, but we'll use A as an example. If you have an A blood type, I don't know what your genotype is, what alleles you have, because with an A blood type, you could either have two A's, so you are homozygous for that A allele, or you could have an A and an O allele, and you would still exhibit the A phenotype because the O is recessive. Again, bit of review. Hopefully that's fairly common and familiar to you. Okay? So DNA, of course, is the molecule that carries the genetic material. For a while in the early 20th century, it was understood that DNA was the genetic material. So basically the instruction manual for how to build an organism. It wasn't until the 1950s that Watson, Crick, and Rosalind, this is why it's awful. This actually illustrates the fact that she kind of got shafted. So I've forgotten her last name. I'm going to look it up here. Franklin. There it is. Rosalind Franklin. So Watson, Crick, and Franklin all should get credit for discovering the structure of DNA. Rosalind was the one who did all of the crystallography pictures. And so she did actually gather the data, and then that data was interpreted by Watson and Crick to figure out the structure of DNA. And it's a fairly complex structure, however, it's repeated. So once you understand the basic units, you know the complexity, and then you just build on it one after the other, almost like adding beads on a necklace. However, we're not going to spend a ton of time learning about all the structure, okay? So you don't need to understand all of it. This is the level that you need to understand. There is a phosphate sugar backbone. We're not going to worry about the structure of that because it's just basically a placeholder. It's the same from one nucleotide or one element of this chain to the next. The parts that you do need to focus on are these parts in the middle. These are called nucleotides or sometimes base pairs. And the reason we say base pairs is because they pair together through hydrogen bonds. So we have cysteine, guanine, adenine, and thymine. Now you don't need to memorize those names, but you do need to be able to recognize A, T, C, and G. We use these letters as an abbreviation for these molecules that can vary along the backbone. Now notice that C and G base pairs have 3 hydrogen bonds, where A and T base pairs only have 2 hydrogen bonds. That will be important later. But for now, just know that there are 4 different possible base pairs and that each one is matched across with its partner. G always pairs with C, A always pairs with T. And so we end up with this double helix where we have 2 sugar phosphate backbones twisted around one another, and the base pairs, which are the actual genetic information. The differences in these base pairs are what cause the differences in the DNA. Okay, so just be aware of that. A chromosome is a very long piece of DNA. You can kind of think of it like a volume in the instruction manual. So humans have 23 pairs of chromosomes. And this will be important here as we look at the amount of diversity that can be created during meiosis. So I'm going to open up Excel to do a little bit of a calculation, and we are going to answer this question. To answer it, we need to know the number of chromosomes that humans have. So we have 23 pairs of chromosomes. They've listed them as 46 total chromosomes here on this chart. And notice some organisms have more, some organisms have less. Plants typically have a lot of chromosomes. Animals, humans are a little bit on the high side, but some animals like horses have many more than humans. So we're on this continuum, and it's not important for different organisms, but it's important if we want to answer this question. What are the odds that you are genetically identical to a sibling? Okay, so to think about this, we need to go back. I'm going to open Excel, and we need to review the process of meiosis. Okay, so again, this should be review for you. You really should have learned this during introductory biology, maybe even high school biology. But I'm going to give you an analogy. Think of it, meiosis, like a dance. Okay. So in these old period pieces, my wife loves watching Pride and Prejudice, Sense and Sensibility, those old British period pieces. And in those pieces, there's often a dance. Now you'll notice that in that dance, people partner up in most of the dances. So a boy would find a girl, they would get together, the dance would ensue, and sometimes there's, of course, important dialogue and wit going back and forth, and that drives the plot forward. But for our purposes, we're just going to concentrate on that partnership. Meiosis, when the cellular division we call meiosis occurs, it's like that dance where chromosome number 1 in humans will find its partner, its pair, and they line up in pairs along the equator of the cell. That's the first important difference between mitosis and meiosis. In mitosis, rather than lining up in pairs, they all line up individually along the midline of the cell. So mitosis is a lot like a line dance, if you will. Okay, so everybody, there's no partners, everyone just lines up, and then the process occurs. But meiosis is more like a partner dance. So that's the first important part to remember, is that these chromosomes line up with their partner. Now, the second important difference, really not critical for this, but I'll mention it now. The second important difference is that there are 2 rounds of division. So in meiosis, after they find their partners, they'll divide, and we end up with 2 cells. Then that cell will divide again a second time, and that ends up making the genetic material divide in half because we had replicated DNA at the beginning of the process. So back to our Excel spreadsheet here. What does lining up with your partner have to do with how genetically identical you are to a sibling? Well, when those chromosomes line up along the midline of the cell, there's no mechanism to have mom's chromosome line up on one side and dad's chromosome line up the other. This is the part that we call independent assortment or random assortment. And so for each chromosome, it's like flipping a coin. When a sperm or an egg is made for chromosome number 1, there's a 50% chance that mom's chromosome gets put into that. Or if we're thinking from the point of reference of the offspring made, it would be grandma's chromosome. And then a 50% chance that grandpa's chromosome will be put into that egg or sperm. So to figure out the number of possible combinations, we're going to use this Excel spreadsheet. So if you had a single chromosome, then there would be 2 possible combinations during meiosis. You could either get grandma's chromosome or grandpa's chromosome, and that would go into the sperm and then make the next generation. And so there would be a 1 in 2 chance, 50-50, that you would be related exactly to your sibling, okay? That you would have 100% genetic identity to your sibling. So this is a rather simple equation. It's just 2 to whatever power, where the power is the number of chromosomes. So if you have 2 chromosomes, it's to the second power, or 4 different possible combinations. So 25% chance that you would be genetically identical to a sibling. But humans have 23 pairs of chromosomes. So if we extend this out, this is 2 to the 23rd power. So there are more than 8 million possible combinations, and this is just for making an egg or making a sperm. So any human can make, because of random assortment, can make 8 million different unique eggs or sperm. Now, the chance of the same egg getting to the same sperm can be roughly approximated by the probability of that times itself. So we're going to do this. So that is 7 times 10 to the 13th power. So if we think the powers, right? So 9 is billions, 12 is trillions. So basically a little bit over 70 trillion. Those are the number of possible combinations that can be made when humans reproduce because they have 23 pairs of chromosomes. So the odds that you are genetically identical to your sibling are more than 70 trillion to 1. That's really an incredible amount of diversity. So think about that. So any humans could have 70 trillion offspring. Any two human couples, they could have 70 trillion offspring. Obviously, way, way, way more than any organism could produce in its lifespan, let alone humans, which have much lower reproductive levels than many organisms. But 1 in 70 trillion and still have every single one of those be unique. Now, the flip side of this is because it's like flipping a coin with every chromosome, you have a 50-50 chance with each chromosome of getting the same one as a sibling. So even though the odds of you being genetically identical are more than 1 in 70 trillion, the odds of you sharing your genetic material you're going to share some material. And on average, you share right around 50% of your genetic material with your siblings. And then the other 50% is different because of random assortment. Okay? Now, there's one other element that you might be familiar with that I didn't even talk about, and that is crossing over. So let me give you an analogy for crossing over, and I don't have numbers to put on it, but it's going to make that 1 in 70 trillion number even higher. So remember our analogy going back to the dance. Meiosis is like a dance where you find your partner. Imagine at the beginning of that dance, you look at your partner and say, "Hey, I really like your arm." And your partner looks back and says, "Thank you, I like your arm too." And you say, "Hey, I've got a great idea, let's swap arms." This is more like Pride and Prejudice and zombies now, right? But you pop off your arm, you give it to your partner, she pops off her arm, gives it to you, and boom, you now are a combination of your original material with a little bit of your partner's material. Now this occurs during meiosis. Chromosomes that line up can swap parts. This is called crossing over. And that creates even more possible combinations and even more diversity during meiosis. So really, it's probably much, much more than 70 trillion to 1. But I don't know, and I don't know that anyone has ever attempted to put numbers on the diversity that's generated. But it's just astronomical. So hopefully, as you were growing up, maybe even today, your mom and dad tell you that you're special. They are right. Even if you have many, many siblings, there's no one exactly like you because your mom and dad are unique individuals and those combinations that are unique. Now, this creates lots of diversity, but it's a diversity through mixing things that are already there. At the element of the genes, we did not create anything new. We just mixed and matched whatever was there. So all of that diversity is possible because of ancient mutations that created different versions of those genes. So just keep that in mind. It's still the ultimate source of all diversity is still mutation, but meiosis mixes it together and creates many, many different patterns and varieties out of the diversity that's already there. Now, we have to go from a molecular level at the DNA to being able to translate that and somehow get that into a phenotypic expression. Okay? And to do this, we use the genetic language. And the genetic language is made up of codons. Okay, so we're going to do some more vocabulary here. Again, hopefully I'll review for you, but it's nice to brush up. Transcription is where we take this DNA code in a gene and we turn it into RNA. It's a chemical reaction. It's called transcription because RNA and DNA are very, very chemically similar. So it's like taking one language and making a copy in nearly the exact same language. Okay? Translation is where we take that RNA, it's called a messenger RNA, and we translate it into a protein. We don't need to worry about the process, that's a bio 1 thing, but we turn it into a series of amino acids, and amino acids are very different than DNA and RNA, which are nucleic acids. And so because of that, we call it translation, because we are now in a very different chemical language, okay? Those amino acids, right, are the basic building blocks, the components of proteins. And proteins are what give you your phenotype. They're what do the work, do the jobs of making your eye color, or being a transmitter, making transmitters, connecting tissue together. If there is a little function going on in any cell, however small or however large, it's a protein or a number of proteins that are doing it. And a codon is the word, if you will, in the genetic language that allows us to go from one to the next. Now, how many codons do we need for all of the amino acids? Well, it turns out, we'll fill this in a little bit as we go. It turns out that there are 20 unique, distinct amino acids that we need to code for. Now, we only have 4 base pairs in our genetic language. So if one base pair coded for a single amino acid, we'd only be able to code for 4 amino acids. Not enough, right? We need 20. Okay, so let's do 2. So if a codon was, come back here. 2 base pairs, we could have AA, which would code for 1 amino acid, AT coding for another one, AC, AG. We could code for 4 amino acids that started with A, and then we could code for 4 amino acids that started with C and with G and with T. So in total, we could do 16 different amino acids if a codon was 2 base pairs long. However, that's not enough, right? We need 20 amino acids. So we need 3 nucleic acids in each of our codons. And you're probably already familiar with this, but I'm getting into more detail and talking about the why here. So AAA, AAC, and then I won't put them all here, but so on down the line. And it turns out that this is 4 times 4 times 4, or 64 different possible amino acids that can be made. Let's do this, put one more down there. 64 codons that can be made if we have 3 nucleic acids in each of our codons, okay? So now suddenly with 3, we have more than enough, right? We only need 20 amino acids, but we have 64 codons. And this is the way the genetic language works. So AAA codes for a single amino acid. Now, this is a chart. You need to get familiar with these charts. And in fact, I'm actually going to replace this one. Probably in the PowerPoint you download. If you downloaded it earlier, you'll see the RNA version. It's a little bit confusing. On your worksheet, which you should actually take a look at, right? Start working on it. Sorry, guys, I've got to log in here. Thought I was already logged in. Okay, on the worksheet here, you've got that same genetic code, but in the DNA version, which is a little easier to work with, and we don't have to switch back and forth. Really, the only difference is it's got T's instead of U's, but it's a little confusing for some students. So I'm going to take a version of this one and we'll put it into the PowerPoint here. Okay. Let's try that again. Copy. Paste. There we go. I don't know why it wasn't working before. Okay. So back to this. There are a couple of ways you can do genetic codes. I like the circular versions. I think they're a little easier and faster to read than the square versions, but you may see square versions before. In the circular version, the inner circle represents the first position in the codon. The middle circle represents the second position, and the outer ring here represents the third position. So if I want to translate AAA, I come to the middle, I say AAA, that codes for lysine. AAC codes for asparagine, and then so on. So you need to get familiar reading this on the worksheet. You'll get some practice doing that, okay? But we can now look at any 3-letter sequence, and we can tell you which of the amino acids. And these amino acids, we've got the full name, the 3-letter abbreviation, and then the 1-letter abbreviation also, okay? So CCC codes for proline. Now notice, if I change, so let's come back here and let's change this instead of AAA. Let's start with CCC. And if I change that third position, so let's say we've got a CCG. CCC codes for proline, C, C, C. So does CCG. So there is redundancy in the genetic code, and I wish they would just call it redundancy, but unfortunately they don't. They call it degeneracy. And degeneracy in common language usually means something else. But in the DNA language, when we say it's degenerate, that means that there are multiple codons that can code for the same amino acid. And that will be important later on and is important for you to figuring out the problem with the genetic code on the worksheet. So the degeneracy of the DNA code happens because we have 64 different possible codons, but only 20 amino acids. Now, 3 of the codons, TAG, TAA, and TGA, those are also stop codons. So those make a signal that's saying this is where the protein is going to stop being translated. Okay, but just be aware that there's degeneracy and that you should be able to look at a genetic code like this and be able to translate codons. Okay, a last couple of terms, I guess, reminders. Again, hopefully you already know this, but if not, and I've used some of these terms already, so this will reinforce it. An allele, again, is a version of a gene. A homozygote is an individual that gets 2 identical alleles, and we will often represent alleles with big letters representing a dominant allele, little letters representing a recessive allele, and you're going to see that going forward in other discussions. A heterozygote has 2 different alleles, and so if you're a heterozygote, those alleles have to somehow get together and decide what your phenotype is going to be. Dominance versus recessive, codominance, so on and so forth. The genotype is the combination of the alleles you have. And we can look at one gene or we could look at all genes, and that's the genotype of an organism. Your phenotype, of course, is your physical expression. We've already talked about this, right? Your phenotype is a combination of both your genes and your environment. Now, regulatory elements are non-coding portions of your DNA that are still important because they help to turn on and turn off they determine the expression levels of the genes. So a regulatory element is not a gene itself, but it's still important for phenotype because it helps to control when genes are being used. Okay? And so finally, last little bit of vocabulary word is this recombination via crossing over. So there are basically 2 forms of recombination. One is random assortment, right? And so we recombine the genetic material into new versions, new varieties by random assortment. But we also have a second type of recombination, crossing over. And that was the analogy we used with the swapping arms. Here's a diagram showing how it occurs during meiosis. And so both forms of recombination create all sorts of different variety and diversity. And that's one of the incredibly powerful things about sexual reproduction, is that we can create massive amounts of diversity, but nothing new at the level of the allele. It's still the exact same alleles that were there before. We didn't create any new alleles. We just mixed them and matched them together in unique ways. And this amount of diversity means that sexually reproducing organisms have way, way more diversity and more possibilities than asexually reproducing organisms.